Okay, I think we are good to go. Good day, everyone. My name is Saba Fanagi. I'm the product manager of Nanocellect products. I'm very excited to have you all guys on board. And today we have Tom Chu with us. Tom Chu is the field application scientist for Asia Pacific at Nanocellect since 2020. Tom obtained his PhD in plant virology, and among all of his expertise, he spent over 4 years working as a principal scientist at Thermo Fisher Scientific. And after that, more than 8 years, he spent leading a protein expression and cloning group at Scripps Scientific Research Institute, and that was before joining Nanocellect. I'm very happy to have you, Tom, on board. Please go ahead.
Thank you, Saba, for introducing me. Good morning from South Korea. So I'm traveling right now, but it's my honor to meet you here and discuss our technology and application. Hopefully, can be in your area. Let me share my screen. Today I'm going to talk about our WOLF and its technology, and so I'm going to briefly introduce our company and our technology, and then give a little bit in-depth discussion in terms of the benefit, how we can use in some of the applications. The application I'm going to touch on is the cell line development and also a genomic application, typically 10x or QIAGEN. So stop me anytime and ask questions. We can hopefully discuss what is in your mind. So Nanocellect is a startup company based in San Diego, California. We are a spin-off technology company from UCSD at around 2000. The technology is exclusively licensed from the institution, particularly is chip-based microfluidic sorting technology. Based on this connection with this UCSD professor, Dr. Low, we licensed the technology. We made the prototype around 2012 to 14, and from there we developed in alpha type and the beta prototype, and eventually leaded to the commercialization of the WOLF around 1928. So actually, our cells mostly take off from 2019. So it's pretty much 3 years right now. We sold about 250 units throughout the world. And the latest version of the WOLF is the G2. Sorry, it's just the WOLF. It's just the Zoom that blocked me some of the portion of the screen. But that we start to deliver G2 since 2021. So far, we have a good sales about that. We're in a series of finance to support our commercial development, but also primarily the R&D one. So beside the first generation WOLF, which is a single laser microfluidic sorting system, now we're into the second generation of the WOLF, which is G2. It's a 2 laser based system. Right now, we are also in development of image based sorting system. That is 2D imaging based sorting system we call Verlo. So that's still in development. Hopefully, it would be come out next year or a little bit later. So that's that's our development. As a technology company, we're mapping into new product and new technology, and hopefully we can do something to be helpful in terms of research for human health, things like that. So that's primarily our development. You would heard this many times. That is the chip, and that's it's the microfluidic chip that is in the heart of the technology, which is showed in this this image. This is a chip, and this is outlet for A and B and C channel, where you have the sample and shields in, and this vent is for discharge of the waste, things like that, and air damper, things like that. So the technology is licensed from UCSD, and it's being supported by NIH grant, 11.5 million. Actually, we're still receiving the grant as of today. We have a 3-year contract from NIH to do more research in terms of developing this microfluidic cartridge for other applications. We did receive some venture capital from well-known companies like Illumina and Agilent Technology. They have venture capital branch of their company. So today we have some long list of publication is on our website, and some of them we haven't been updated yet. But we also have therapeutic products in phase trial. For example, this from Crispy therapeutic company of this VCTX 210. So we're making progress into many areas, including some of the medical areas. If you want to know our company in an overview, and this is some of the major customers over the years that we have been able to place our instrument to. So you can see there's a big pharma, like Takeda, we have even 5 units there. Roche, we have quite a few. Small biopharma companies, they're doing all kinds of pioneer scientific research in their area, and contract manufacturers. So those are companies that are doing service for other companies for particular, say, cell line development or collect cells for genomic applications. Academics, there's many well-known names. I don't want to, but you can look over, and some government institutions, including government-sponsored national research institutions. We also have a good coverage in Asia Pacific. So that's the overall our customer base. This is just some example. So as I said, we have over 200 instruments throughout the world. We are really emphasized on service and support and try to make customer happy and success. So this is our field application teams, which is meant to help you to understand the platform and doing demo and application support. And this is our sort of R&D research, just doing applications for particular areas that are concerning tech support teams and customer service success team. We have over 36 application nodes on our website right now, and we have multiple protocols, including the one that you're concerned about is the cell line development and also the genomic sample preparation protocols. So those are our general resources available to support our technology, support our customer base. Now I'm going to switch the gear to focus more on the technology and the platform that we are use. So this is the WOLF. WOLF is a reverse of the flow, right? WOLF. It's a single laser, 488 laser, which is the most popular ones. and it can detect two scatter, forward scatter, back scatter, and up to 3 colors, FL1, 2, 3. That's basically green, yellow, and red. Those emissions that we can detect. So by WOLF. G2 is the next generation of the microfluidic sorter based on the WOLF. It's the same principle, same procedure, except we add one more laser. Of course, we optimize some of the internal component to make it more reliable and user-friendly. But the other laser could be, in addition to 488, could be 405, 561, 637. So in other words, if you are using DEPI or M-Cherry or APC, then those choices are probably good for you. For cell line development, we normally dispense them into 96 or 384 plate with the N1 dispenser. So this dispenser is work with WOLF and the G2 to be able to do plating for single cell cell line development. I want to point out that the reason, particular in terms of cell line development, why it's good is that we have a system that you can sort and you can dispense, right? So that's one system. And for cell line development, the beauty of this is small, they are small enough to put it into biosafety hood, where you can operate the whole thing in a aseptic way. Another benefit is, beside the microfluidic, which is pretty gentle, is that we can use your cell culture media for the shields, for a flow and sort of your sample where you put it here. That is particularly if you're using sensitive cells like neuron, stem cells, things like that, protoplasts. You do want to use optimized media for them to make them happy, because otherwise they are easily die or into a non-optimal situation because of the stress. The whole thing is controlled by WolfView software. The software is pretty simple. You just can gate and sort. And there's some settings you can use, but the overall flow is from top to bottom. You just follow that. Yeah, that should do it. Because we're talking about two topics. One is the single cell sort, the other is cell line development. We do have two type of cartridges. They are fundamentally the same. You know, the structure are mostly same, like PCT, right? But the bulk saw compared to the single cell sorting has extended tubing to be connected to the dispenser, the N1 dispenser, for plating into 96 or 384 wells. So that's pretty easy to understand. Where the genomics, you just enrich or collect some samples, you can simply use the bulk sort. For that, we have two type of cartridges. That's just for your information. So we think we are new technology. We are developing this product with the hope that we can overcome some of the issues associated with traditionally sorter. For example, the droplet sorter. For example, the BD area, right? They're quite bulky, but they are workhorse in terms of the core centers. But they are high efficient, but they have some drawbacks. Those are the things that when we initially design our WOLF platform, those are the things that come onto our mind. Typically, the poor sterility associated area, because area it's very big. It's an open system, right? There's some seals, but they're generally it's open in the open air. Poor sterility, long wait time in core center, inconvenient travel because you have to travel there from your own lab back and forth and bring the samples, have to be cold, things like that. high cost of maintaining the facility, no efficiency in terms of single cell dispensing, and this is more technical, no efficiency, or talk about why it is no efficiency, primarily due to the high speed or the high pressure coming from the design of the sorter. Difficult to use. It is difficult is because the software and the operation are very difficult. It needs a dedicated technician for dedicate that service, and it's pretty high maintenance in terms of get rid of clog and things like that. Dead and dying cells, high debris samples. So because the high pressure, sometimes the cells probably just sorted, it will be alive, but over time it die very quickly because the high pressure. So our wolf would have the opposite of the traditional droplet-based sorter. The biggest outstanding difference is that we sort things gently because we use nominal flow, right? It's a microfluidic sorter. It's plastic chips. I mean, you can see it wouldn't be able to stand too much pressure. But the nominal flow itself, luckily, don't require that much pressure. It's typically less than 2 psi, or we normally just compare to human blood pressure, which is around 2 psi. So that's how the cells endure during the sorting process. Small footprint. It's fairly small, and typically you can see from the picture at Millennium, it fit in the hood and fit in very well along with N1, and it can withstand UV. So for cell culture people, which I used to be a cell culture scientist, and you do want things to be sterile, to be very concerned in terms of that, because even though that is the most simple thing you can say, but it is actually most challenging thing for cell culture people. So another thing is no aerosol. Because of low pressure, it wouldn't generate aerosol. Aerosol require certain pressure to generate. And simple and intuitive software. When we do the demo, you would understand. It's pretty simple, it's streamlined, and you can use it in one day. By yourself. Fractional price, of course, will log a scale difference compared to BD area. So this is our introduction video for the WOLF G2, which is the next generation WOLF. This is how you put the plate, dispense the plate, and operate the G2. The primary characteristic still is gentle and it's user-friendly. So I want to basically introduce some of the technology used so that you can understand it a little bit better. WOLF or G2, they all look like that. So this is a sample tube and this is the shields. As I said, you can use your cell culture media for the shields, and those are the pumps. Pump those into the cartridge. The cartridge, there's a tubings connected to the dispenser for the N1, but most importantly, the fluidic flow into the cartridge, and the cells also flow into there. Because of the shield's flow is quicker than the cells sample, it form a beamline, and where it can be sorted based on the PZT action. I will show you a little bit later. And it's got dispensed into the 96 or 384 well, where you have the cell cultural media already dispensed there waiting for the cells. And after dispense, you can just bring this out, cover it, and put it into the CO2 incubator for cell culture and for the clonal outgrowths. This one, if you have questions, please ask me. I hope that we can have a discussion on the technology and so on and so forth. So in the heart of the technology is this so-called PZT, piezoelectric actuator. When the electricity applied to this metal thing, the physiological property of this metal would be able to either bend or push it up. So you can imagine if the cartridge, this cartridge is primed, so the liquid would fill in all spaces, and where the cell form a stream because of the shields from the two sides, and it would flow here, and the laser would hit the sample, and the emission get be connected, and the software or you decided whether to sort it or not. If you want to sort it, and the PZD can generate the electricity pulse so that it's either bend or pushed, either go this way or push out this way. So if it's go this way, it pull the cells into this channel. If it's pushed, because it's all filled with air, right? There's no void space. It would push the cell out there. So that would be sorted into A. So that's how it's being sorted, and then flows through the system into either A or C. So there's two channels can be sorted, and the flow through would go to B. Like if you have precious samples, you collect it to the B, and you can reload the sample to be resorted into A or C. So this is a sample is pretty precious, and this is the prototype where the door is not fully established, where you insert the cartridge there. And as soon as you insert the cartridge, the PZT get engaged with the ping from the wolf, and so the action can be started there. And so that's a very simple physical sorting mechanism, and that's how it works. This is just a cartoon to show that the cell stream, because of the PZT action, can be pushed here or pulled here. But of course, in this case, is pushed here. This event can happen 300 times per second. So in other words, the 300 cells can be sorted per second. That's not quite slow. So it's a very fast process. As I mentioned, both bulk sorting and single cell sorting are done through the cartridge or the chip. The chip comes with in a pouch. It's completely sterile inside by the ethylene oxide. So if we said the wolf or G2 can fit in the hood, but if you open the pouch, the cartridge inside of the hood, you are golden in terms of aseptic or microbial contamination, because everything is sterile. So that's why this whole thing is fit into cell line development very well. Where BD you cannot imagine, or other platform you cannot imagine you can do that. Another thing is because of this is disposable cartridge. So in other words, the setup might take some time to prime and things like that. But after you're done the sorting, this whole thing can be taken out and disposed directly into the biohazard waste. And because the cells wouldn't get into the machine, it just get into the cartridge. So that's one of the actually one main advantage of our platform. Now, when we talk about the cell line development, the traditional sorter can do that as well, right? I mean, they are a sorter, but this cartoon to show that the traditional sorter, even though they can do that, but to form a droplet streamline, they have to use the pressure close to 7 psi, 70 psi. That's pretty high. And the cell got squeezed very much. But when they come out of the nozzle, there's a decompression shock, because in the air, there's no pressure, that much pressure. So the cell got busted or easily, even though you sort it after, it probably intact, but afterwards, a few days later, they could die because of the injury generated during the process. Same thing, some other mechanism of droplet-based sorter, like jetting air, except the laser position is a little different, but the pressure is still substantially high. So in this case, close to 60 psi. Ours is using, here's a WOLF using nominal flow. The shields coming from two sides, some sample coming from middle. But the cell stream formed is still pretty well, but they wouldn't endure that much pressure from the traditional droplet-based sorter. So this is how it's being sorted, laser hit it, and interesting, the cells get sorted into the channels that is designed. The main benefit, again, is the low pressure and the gentleness toward the cells. There's only 2 psi to change from within the system wear out. So that's the advantage of our system. Any questions? Okay, I will just continue to drive this one. Because this is experimental data to show, give me a second, let me move this a little bit toward it here. Okay, WOLF has highest post-sort viability. This is 4 hours after sort. Ours is not too much difference than some others, of course, better than some of them, more obviously. But after 4 hours, not too much different. 24 hours, you can see ours is staying at 96% viability. And with 96 of them still viable, but many others already decreased to 90%. So that's one of the things that the low pressure sorter can be beneficial in terms of cell viability, particularly when you do cell line development. That is something you want to consider. Okay, so we're gonna drive into the cell line development in this application. Before we talk about some examples, I want to mention the traditional way of doing cell line development, which is through limiting dilution. The idea is that you genetic engineering cells in your population, and you dilute the population to the point that in each well of 96, say, you can dispense the volume so that there's 1 cell per well, and hopefully this cell going to develop into the colony you want. And of course, you keep track of this by doing imaging. The downside of this, the manual, the limiting dilution is pretty simple, right? I mean, you just do the dilution series, but actually there is some downside beside the huge amount of label and the imaging of the plate is that you do the dilution, you try to get 1 cell per well, but because of cell-cell interaction, there is a lot of doublet or potential doublet. So ideally, even though you got a colony, they might not come from single cell. And your goal is to get a single cell monoclonality of the limiting dilution. So what you do is normally people dilute it more, try to still focus like say 0.5 cells per well. And the thing on that is a lot of doublet get decreased, but a lot of empty wells is also generated. Of course, among that still single cell there. And some people even dilute it more. Overall, the process is not efficient. Typically, we see anywhere from 19 to 30%, depending on the many factors, particularly the media as well. The cell sorter, when cell sorter play into this same procedure, the thing that cell sorter is different from the limiting dilution is that the cell sorter is a machine that can see the cells in terms of size. It can differentiate it from debris, right? And also differentiate from dead cells by doing some dead and live thing, which is typical in terms of flow cytometry. And then, of course, if you have GFP or some other fluorescent marker or dyes, and those can be easily recognized by the sorter. Because sorter is first has to be a flow cytometry to begin with. So that's why adding this instrumentation in this workflow in terms of cell line development, it will greatly enhance the efficiency of the cell line development. So that's the whole thing. I mean, you can do cell line development by your manually, but if you have this wolf for this purpose, it would greatly help you to increase the efficiency of that. The wolf N1 can have, for live cells, for example, CHOK1 cells can have 90% deposition rate. That the deposition means 90%. That means that in 100 wells, there will be 90 of them are single CHOK1 cells. That's pretty efficiency. This is 94, and this is 384, and this is confirmed by imager. This is to show you that in a 384 wells, if you use wolf, you can get this type of the colony growth from single cell, and that's pretty efficient. And we have multiple data to show that we can reach 86% in terms of 96 well for the monoclonal growth, and 384 a similar number. So that's pretty high in terms of efficiency. You would never get this high in terms of limiting dilution. One of the key things for cell line development is monoclonal growth. You have to demonstrate that. This would require image to follow the single cell deposition throughout the day 0, day 1, day 2. Typically, we done day 7 and day 14. You need to monitor it. It's from single 1 to 2 to 4, things like that, to be compliant with GMP, whatever you're using. So this example to see cell line development where limiting dilution at different dilution ratio, you would have a different growth ratio. But if you use wolf, it would be reaching 61%. So it's like the average of this is probably more than 5-fold difference. And this is from another company, life science company, where they are using limited dilution at 0.5 cells per well. The efficiency is around 5%. We're using ours, it could reach 45%. I have to point out though, you can see this number is everywhere. In other words, it depends on the cells you use. Is this HEC cells, is GUP cells, it is suspension cells, adherent cells, or some other cell type. They all behave differently. So the comparison, so that's why we refer to limiting dilution or other type of the sorter. But anyway, the difference between limiting dilution and our sorting into the 96 well plate could be like a 48 fold difference. Let's say 5-fold. In other words, you have five plate using limited dilution, and you could just use one for our from our platform and reach the same clonal outgrowth. We use this slide to say that using our platform, you would save a lot of space in terms of the CO2 incubator space for the plates. Because if you do limiting dilution, you have to culture those plates, and also you need the imaging every one of them over time. So by saving plate, increase the efficiency of monoclonal growth, you save a lot of plate, culture space, and imaging time, of course, and then labor. So that's one of the things that we say this could justify your purchase. I move into another example now. Now I'm talking about CHO suspension cell. This is one of the slides happened during our optimization of the CHO suspension cells for monoclonal growth. In this case, we only sorted based on the forward scatter and the back scatter. And you can see at day 7, there was a lot of colony growth. And at day 14, the colony became bigger and recognizable on the microscope like this. So it's a typical coming from a monoclonal growth in terms of CHO cells. Some of the customers using this without culture optimization, the media optimization, they would get very bad result. So this is how we improve from those experiences by optimizing the culture media. in this typical case, we reach 91% of monoclonal growth, where there's some still have two colonies in one well, and there's some there's no colonies in one well at day zero. So, but overall, we can reach 90% of the monoclonal growth by using WOLF to dispense the CHO cells, just based on the forward scatter and side scatter, or just the cell itself characteristic, not even the dead and live dials. And this is some of the compare with some of the our close competitors. Those data are from their website. Because I was part of the effort to develop the CHO cell outgrowth for cell line development. So I know that those are from the website. We have those data if you're interested. But regardless, it may not be apple to apple, but compared to say other CHO cell lines or from other brand of the sorter, like from Nanocell, J's or cell printer area, things like that, ours outgrowth, clonal outgrowth is a lot better. You know, we are optimized in this application, not only the workflow, but also the media as well. And we have established protocol on this. I'll be more than happy to show you some of the publications we have in this area. I will mention that either probably after demo or something, some of the publications we have in this area. The summary is that our microfluidic sorting increased the quantity and quality of clones compared to limited dilution. WOLF and G2 are optimized for CLD development. That's what I'm saying. You know, we can put in your hood. They can withstand UV radiation to be sterilized and work with N1. And also, you know, you can use your choice of cell culture media for shields. And we have a number of ways to treat the samples, use certain sample concentration for optimizing monoclonal growth or sorting. And we'll work with a software that is, you know, has a user interface that would be obvious to you when you use the single cell dispenser into 96 or 384 wells, and stay sterile with the microfluidic cartridge. This is common with support, and everything inside you can be guaranteed that they are sterile. So if you open that into the hood, you get the culture medians sample in the hood in a sterile environment, you would be golden without no worry about the contamination. Let me know if you have any questions here. I may not explain to you very well, but we're gonna move on to genomic sample preparation if you don't have any questions. Okay. Before I want to talk about the genomics, I want to point out though, for cell line development, we're dealing with live cells, and we want those live cells to be able to grow into colony, right? That's the goal. But for genomics, for example, 10x, I would just use 10x. We're gonna bulk sort. Okay, first we bulk sort them, we collect a bunch of cells. We want those cells alive, right, during this process. But then we process them to extract the DNA or RNA. So the cells are done with that part. The reason that we want those cells to be live so that hopefully they maintain the integrity of the cellular functionality in terms of the RNA transcription, translation, things like it's not being altered too much because of the cell dying. So that's the purpose. You know, we're trying to get the live cells extract their authentication, the DNA or RNA profile, and be sequenced to reflect that stage of the cell health or their transcription or translational status. So that's kind of the purpose. In terms of that, we use, of course, in this case, we use a bulk sorting cartridge instead of single cell cartridge, sorting cartridge. So the WOLF in this case is part of the tool in terms of sample preparation. Okay, it's prepare a good sample for Chromium and then be sequenced. So that's the idea out of it. We have actually dedicated scientists to do this for quite some time, and we know exactly specification and protocols on this. We need a good like 10K cells to be able to do a good job on Chromium. So we have a lot of that on our website and in our publication. But for me, I'll just briefly introduce some of the benefit of using a sorter for sample preparation and then for your genomic application. Of course, people probably know that the genomic application C10X are quite expensive. So the idea is that you want to get a quality sample before you load the sample on Chromium, because otherwise you could waste your money. In this example, we just use a cartoon to say like this is the cells you want to sequence for information, right? For DNA or RNA information. But there are something like about to die, the cells, or the cells are already dead. Lots of things start degrading, or the RNA get chopped off, things like that. So if you sort, you could get 90% purity of those target cells, and they are viable, they are kicking, and then you will get the right information. But if you don't sort, let's say this is a sample prepared, you get your best effort. You go to Chromium, and half of them would give you the information. Actually, those other half would just make you crazy because they're noisy. So that's some of the ideas. This is just another example. Like, what about mixture of cells, C and B, and this is the A where you're interested. So you also want to sort for the A. You enrich it and you put it in Chromium, you get a lot of information from A. That's what you want. So that's the idea, is that why you want to use some kind of cleanup process for your sample preparation. And that's the whole thing, that why WOLF or G2 can play a role here. This is just to say that in your sample prepare, you have all the debris in the dead cells along with your target cells. Now, if you sort to get rid of the debris, which is this typically any sorter would be able to do that, right? Pick up the live cells. And if you use this sample for sequencing versus this sample sequencing, the dramatic difference is that this is mostly our target cells. The debris and dead cells are minimized on this post-sort sample. Same thing here. If you do that is debris, right? Dead cells. But if you want to get most of the viable cells, you can stain the dead cells and sort for the viable cells. And this is the one that you could use for sequencing. And that would be great for you, because most are dead cells. Most are live cells after sorting. This is just another example. We use PBMC unsorted versus sorted, and on Chromium, and to show the data differences there. I just want to point out that some of the typical standard that people look at, like median UMI count per cells, the sorted one significantly increased over the unsorted one, say over 300%. There is something like median genes per cell, meaningful genes per cell. So that's also significantly increased versus unsorted ones. Typically, this is what the sequencing folks would look at, the t-SNE plot. You can see this is an unsorted sample, and those two are sorted sample. I sometimes look at the color of the cluster. You can see the projection of the cells, but this is the UMI count. And on the unsorted one are pretty blue, I mean, in dark, right? Where the sorted ones are much more lighter, even in yellow range. So it's much more enriched or more UMI count in this one. People also notice that here there is 7 clusters, 9 clusters, where the unsorted one only have 7 clusters. And plus the cluster itself is well separated, where this one are almost like a cluster together. So the quality of after sorting for sample preparation for genomic sample preparation is pretty useful, meaningful in terms of research to simplify and speed up your research. This is another example of 10x application. In this one is the iPSC cells being used. So sorting for the most prostate cells for the scRNA sequencing result. I'll just go to the result part, and this is the graph to show some of the typical markers or flags for the comparison of the sample quality or sequencing sample quality. This one is the medium gene per cells. The WOLF sorted compared to unsorted and the area sorted are showing increase in terms of many genes per cells. Now there are more information generated per cells, and other typical count would be the UMI count. You can see that the WOLF sorted are still good. And this is the opposite one to indicate RNA contamination. The fraction rate not in the cells is from other contamination source, where the WOLF sorted are significantly lower. Estimate the number of cells recovered. The WOLF is kind of matched the expected one. This is some of the examples we got before. This is some of the pictures that we got from a lab, though. Typically, people have cells to count it and to understand how much cell concentration is, and you can prepare the cells to be sorted, and after sort, you can put it into Chromium. So you can see the lab space is really small, and with that, you can get really good data out of it. And there's a lot of customers are using this technology. This is some of the names. I would say today we cover 2 topics. Single cell line development. Cell line development could be CRISPR, could be iPSC, could be CHO or hex cells, or genetic engineered construct or whatever. So there is a broad range of the cell line development. Another part is the genomic applications, which is to get a good sample preparation for sequencing. So this 2 market is from our understanding so far, is about 80% of our customer space. 80%. So that's a great part of our product where this is a sweet spot. So the WOLF in a cap, so it's a small, easy to use, and gentle for the cells to give you highest viability. Of course, it's safe if you're doing microscope microbials and or HIV or lenti-infected cells. Low cost is a fraction of the cost compared to traditional droplet-based sorter, and it's meant to put it into your hood for cell line development, or if you just do the genomics, put on bench for day-to-day use. It's not for a dedicated facility or people to operate it, to monopoly the operation. So that is my presentation. If you have any questions, please let me know.
Do we have any question from audience? I have one question, Tom, for you. Is there any limitation on the cell size and user can sort on the WOLF?
Yeah, so like typical mammalian cells, it would be within our range. But let's say if you're running some protoplasts, the cell size could vary a lot. Our COC cartridge, the maximum diameter would allow you to sort up to 65 micron cells in diameter. So that's actually pretty huge, but probably not huge enough for you to sort organoid and things like that, or some other crazy cells. But typically, anything from bacteria, yeast, some microbia from ocean, fungi, or Caldiamyces, haplotype, neuron, stem cell, would muscle cell would be all typically be fine.
Perfect. Any other question from audiences? Janice has a question.
Sorry, I'll just shout up. With the G2, you have the choice of a couple of different second lasers. Are there any plans for having a system with 3 lasers?
Not at this moment. Based on our survey, we would think that with 2 laser system, particularly the second laser, you could pick from 405 or 561 or 637. We would think that we would occupy 70% of our market. But of course, if you have any applications, we'll be more than happy to look into that. But at this point, beside the forward and backscatter, we can detect up to 9 colors, and that 9 colors is actually take a lot of control to run. So, but yeah, we're thinking about that. We haven't decided yet. Yeah.
Thank you.
One more question that I received from many customers, Tom. How long each cartilage can be used, and is it possible to use it at different cell types, that you run the samples of different cell types on one cartilage?
So typically, one cartridge would last 6.5. That's our official quote, okay? Typically, you can run a full day with that. Now, if you do cell line development, your cells is so precious because your cell line development take months to develop, and then you characterize and probably take half a year, even a year. Do you want to mix with other cell types? You can decide. But if you're doing like bulk sorting or something, you can do flush in between, probably get a good and clean sample prepared for the next one. I would say like, say you need a 10K cell for one sample. 10K cells is at 200 cells per second is maybe just 1 or 2 minutes, right? Yeah, so 1 or 2 minutes you would be able to get that many samples. So for 6 some hours, typically is a one day. You have many samples to be used on a single cartridge. Is provided you flush in between. You know, there might be lingering leftover cells in the caveat or somewhere, but majority for people doing multicolor panel, they need different colors and you need to analyze each colors, and then to the point that they eventually analyze their sample. That should be no problem. So I would say, you know, you could use quite a few samples for single cartridge.
Beautiful. Okay, if we don't have any further question from audiences, I can just showcase our demo here in our workshop at Millennium Science Headquarters in Melbourne. Of course, we don't have real samples to run on the instrument, but after I finish with the calibration of instrument, Tom would be happy to show you some real data on the application he covered today. So I would ask my colleague Janice here to help me out with trying to show the WOLF steps, automated steps, and also the software that you will see shortly. And feel free to ask any question during the time. I've done some preparation beforehand to just save the time, and I know all of your time is so precious. So feel free to raise any question during the procedure. Well, okay, that's the cartilages that we talked about. Microfluidic cartilage look like that. I'm gonna open that and show the cartilage to you. This is a bulk sorting cartilage. Also, we do have a single sorting cartilage. If you would like to do the single sorting, the cartilage look like this, coming with the input and output tubing. And this gold microchip here is where the whole technology relies on. Okay, I'll undo the tubing. I'm trying not to touch much, and I'm not supposed to touch the glass side of the cartilage. So to start our work, I need to slide this cartilage into the system and open the door. Here is our cartilage holder, and this is the lash on top of that. First, I need to make sure that our software is connected to instrument. Probably because you guys don't have the best view of our software, I'll ask my colleague to bring our camera a little bit closer at this point, so I'll be able to show you the whole workflow here. And through my demo, I'll try to explain better so you have a better understanding of how the WOLF Viewer software look like. On the left side of our screen, we have all our automated workflow, and these are where we are focusing on, and I explain further when we go through. Here is our data plots that we can go through it when we analyze our calibration beads or our samples. We'll work on that plots. And also on the other side, there is a green button here that tells me that my WOLF Viewer software is connected to the instrument, and it comes as a green light here, which tells me that the software and instrument is connected. Okay, we put back our camera, and through the steps, I'll take you through all the notification so you have a better understanding of what I'm gonna do and what type of a notification you'll receive on your software and screen and what you need to do. It's pretty straightforward, and it's a very easy and friendly way to work with that software. And technically, there is no way to muck around. So first step is to put the new cartilage. On the top, it tells new cartilage. If you're able to see that, it becomes like a darker. I click on new cartilage on that top left, and then it asks me to make sure that the cartilage present there, and then press OK. So I press OK. And the instrument, as you may hear the noise, start the motor initiating the workflow. And then when that has done, it tells me to insert cartilage now and press OK when done. So I will insert the cartilage here. And this gold microchip needs to go first this way. And there is no pressure or anything required. You just need to smoothly slide your cartilage in until you hear a click. So I hold the lash up and then slide my cartilage in until I hear the click. You heard that? So I put down that lash and the cartilage is okay. So here, come back to my screen. Here there is a notification that you may not be able to see due to lighting, but I press OK here. And at this moment, the system asked me to provide the ID code or barcode code of cartilage. Janice, if you're able to read that barcode code from the side for me, that would be great.
020-326-080.
Okay, I've entered that. And our motors are moving, and the system asking me to make sure door is closed, which we do, and press OK. So there is a system check now that we are waiting for that. So the next step is to prime our instrument. For priming our instrument, we need the filtered PBS, which I have done it. And we also need to fill our priming tube with this filtered PBS. I got the notification on my screen asking me what type of the cartilage I'm using. So it's either can be a single cell or bulk sorting. As I mentioned, today I'm using a bulk sorting cartilage for our demo. So I click on bulk sorting. So here on an instrument, I just removed this plastic bag so you can see that better. On this side is our sheet holder, a 50 ml Falcon tube filled with filtered PBS, deionized water, FBS, or any media that is essential for your cell work should be fine. Maximum for a day, we require 50 ml of our sheets. Today I've used filter PBS, as I mentioned before, and this sheet goes to that sheet holder and stays there. Here is our sample holder, which you can put fax tube or even a pen drop tube is fine. I've got fax tube today. And for priming step, we are, as I mentioned, fill our fax tube with the PBS. I put that here and put my sheet PBS here. And then I need to connect the output tubing to our instrument. So we do have 3 tubing here. Our priming tube, which has a valve on it, our sheets tube, which is the longest, and our sample tube, which is the shorter one. I'll show you how to connect them in a moment. And I also want to connect our sample holder here, which I'm gonna grab that from our box. This is the waste holder. If you're doing real work, you have the sorting tubes that can be attached to here, same way as waste holder. And we have the channel A, B, C here to collect your samples. And your samples can be collected from channel A and channel C. And the middle channel, which is the channel B, is unsorted samples. And as Tom mentioned, if you've got precious samples, you can collect those one and resort them on your instrument. So the tubing, the longest tube goes to our sheet, and there is a pump here. So this tube goes around our pump, and there are stopper around that, which tells you where you have to position your tubing. Both of the stopper sitting nicely down the pump, and then this tube goes to our sheets. when it's nice and correct in a position, I can close that and you should hear a click. And then our sample holder can go to our sample tube the same way as our sheet tube. Two stopper goes all the way around our pump. There is another stopper there. Then it's nice and correct position. We close our pumps and there is a click. As I mentioned, I assume I've mentioned that, but excuse me if I forgot, everything is sterilized in our workflow. And this instrument can go to your hood as well, and then that would be 100% sterile. And the tubing coming out of our cartilage batch is completely sterilized. But if I accidentally touch any of the end of tubing, I have the ethanol wipes here that I'm gonna wipe out before putting the end of tubing to my sheets or to my samples. And one thing you need to remember is that the end of tubing should not touch the end of the your falcon tubes. So you make sure that they are sitting on the right position. And then I have the priming tube here, which is a bigger tubes here. And that goes to one empty fax tube that I'm gonna place here. And this tube will sit here nicely. And the valve, if you can see, there is a valve. There is a white valve here. This valve needs to be open at this stage, and we need to close it after we reach about 90% of our priming process. So everything is connected now. I start to prime the instrument. And here, after the second button here on the left side is a priming. So I click on prime, and then it asks me, the system, that makes sure the tube containing PBS and it's in place in the tube holder, which is there. So I click OK now. And then, as I mentioned, there is a notification here asking me to make sure the purge valve is open, which is open. So I click OK again. And as you can hear, the pumping start to work and priming our instrument. So that normally takes few minutes, but what we can do during that procedure is to go to the third step, which is alignment. So I will go to my software on the third button here is to open alignment. I will click on that and start to align my laser. So then there is a new screen coming up onto my software. And this is fully automated. You don't need to do anything. You just click on the align button here, and the instrument automatically will align your laser. So the goal for alignment is to focus on Z and XY position on your cartilage, which is where your microchip is, and then cells through the magnetic field get sorted into two-way. While we are aligning our laser, the priming is also in the process and it's in progress. So we should be able to finish the alignment soon and go to the next step. I would like to add something here that in the sample folder that we filled with PBS now for priming step, you can add your samples as minimum as 100 microliter. Which means that even with a very small amount of sample, you can sort your cells. And also because it has an open access to the sample tubing, if needed, you can add samples as you go through your step. And if you feel you require to add more samples, you can do that. In terms of if you decided to use media for your cells, if your cells are very sensitive and you prefer to use a cell media as a sheet, then I believe you don't need more than 8.5 ml per hour for running the instrument. So there is a big saving on your media as well when you're running the sheet on your instrument. So alignment is completed now. It comes with the notification on the screen. So I click on OK. And then here, it's very good to scan for bubbles. So you want to make sure that there is no bubble in your cartilage. And if you see any bubbles in your cartilage here, then it's better to go back to priming and reprime your instrument again. So here on the same screen, there is an option for scanning for bubbles, which I click on that and I'll wait to see if there is bubble or not. And our priming step here is also at 75%, which we are still waiting for that. Oh, there is a heavy rain here down Melbourne, so I do apologize if you have any feedback noise for that. Okay, we reached about 90% of our priming step. Now the instrument asked me to close the page valve. If you remember, our priming tube had the valve that we kept it open. Now the instrument asked me to close that. So what I'm gonna do is open the door and physically close that, and you should be able to hear a click. It's a little bit hard valve, so you need a little bit of pressure to close it, and you hear a click like that. So I close the door and then come on the screen, and because I've done that, I press OK. And our instrument is still looking for a bubble. I couldn't see any bubble with my eyes here. So you should be good to go. And our priming is at 98%. So I should be receiving a notification that priming has been completed shortly. So the check for the bubble was okay. So I have closed that because I've already done that. So I closed that screen, and then the next step is to calculate the offset, which is again an automated step. Instrument asked me to ensure the door closed, which it is. So I click on OK and then start calculating offset. It shouldn't take more than a couple of minutes. So after that step, we need to remove the priming tubes and replace it with our calibration bead and calibrate the instrument. So calculating cartilage offsets was successful. This is what instrument tells me. I click OK. And I think one thing that I probably forgot to mention during the priming step, when you want to close the purge valve on your priming tube, when the instrument asks you to do, it's good to check for the bubbles and make sure the bubbles coming through your cartilage and through your priming valve before closing that. I've saw the droplets there, but I forgot to mention that to you guys. So what I'm gonna do now is replacing the priming tube with our calibration bead. So the calibration bead comes from Nanocellect, and it is a droplet bottle, look like this, that we need to bring it to room temperature 10 minutes in advance and spin it for about 10 seconds. So make sure the context is fully mixed before adding it to PBS. So I've got my vortex here. I am going to vortex it for 10 seconds. Okay, I've measured 650 microliter filtered PBS in a Pendrov tube beforehand. And I'm going to add 1 drop of our calibration bead to that and vortex it again. So 1 drop is enough. And close that, vortex it for 10 seconds, and put it into our sample folder. And as I mentioned before, just need to make sure that the tubing is not touching the bottom of my Pendrov tube. So I put it here. I'll check that is sitting nicely. I need to bring it a little bit down, which is perfect. I close the door, and coming on my software, what I can do is to analyze my calibration bead first and then calibrate, which I'm going to do that today. So on the left side, on the analyze, I click on that and asking me to make sure the door is closed. Okay. And now the instrument asked me to name that sample because it's a calibration bead. I will call it calibration. And then because it's a new sample, I call it new samples, click on new sample, and then the instrument start to analyzing my sample. For the calibration bead, the aim is to reach 20 events. And when we reach the 20 events, counting 20 events, we are good to start calibrating. Now, to see more details, I open the data plot here, clicking on this side of a screen, and click on 2D and 1D plot. I'm waiting for fluid to stabilize and then be able to click on that. And one reason that we vortex harshly our calibration bead, because we want to make sure that it's completely mixed with our PBS, and it's not sitting down the ependraw tube. As you can see, the instrument start to analyzing and counting our beads, and we are at 11 events on this side of the screen. It shows me that we are at 11 events per second. So for this one, we aim for 20 events per second. When we reach 20 events, I will click on calibration button here. 17 events. And as you can see on the 2D plot, we are seeing the density plot. On the density plot, our calibration bead, and the peak here on the 1D plot. So we reach 20 here, which is the time to click on the calibrate. And the instrument asked me to load the solution into sample tube, which we have already done. So I click OK. And now the instrument start the calibration process. And it will goes to channel A of the cartilage and channel C of the cartilage. And we need to wait for that process. And when it's completed, we are good to go. The reason that we analyzed our calibration bead, because we wanted to make sure we have enough beads to calibrate our system into that standard. So if we're 100% sure that we have enough beads, we can also go to a calibration step, but this is what we suggest end user to do first. So right now, instrument calibrating to channel A, and after channel A finished, it will goes to channel C, and after that, we can just go to our samples. When you're analyzing your calibration beads, sometimes the signal is low, then your instrument will give you a notification that your signal is low, and then you may need more concentration of your calibration bead. So this is something to bear in mind as well. Calibration to channel A successfully has been finished. Now start to calibrate into channel C. All this notification coming on your instrument, so maybe you don't see that very well, and that's the reason I'm explaining to you, so you have a bit of understanding what is happening on my screen. Maybe, Janice, if you can bring our camera a little bit closer at this stage, so researcher can see what I'm talking about. These are all the notification. As you see, this is calibrating to channel C, and we are 40% of progress at this moment. When this has finished, we are able to flush our beads out of our cartilage and run our samples. Today, we don't have a samples, so we will stop at that stage, but then Tom will be able to show you some data from the application that he spoke about today. Thank you, Janice. Perfect. The calibration was successful. And now the detection setting changed to cells automatically. And then here on the right side, we have a detection setting. When the calibration was successful and finished successfully, then the instrument automatically changed that detection setting to cells. If in case you would like to run a rainbow beads, for example, we do have rainbow beads, you can come here on the detection setting and click on the rainbow, and then it will be change the parameters here. You can also manually change that, but you don't need it because all of them automatically preset for the end user. So the notification tells me that the calibration was successful and setting changed to cells. So I clicked OK. Because I don't have cells, I will, if for example, I had cells, I need to flush my calibration bead out of the cartilage and then replace the calibration bead with samples, of course. But because I don't have the samples, I still want to go through flushing step and shows you how the flushing look like and finish up with the cleaning of that instrument. So to show you how quick and easy this step is. So I will come here and remove my calibration bead from the sample holder and then replace it with the filtered PBS and flush the beads out of my cartilage. On this side, there is an option to flush. I click on flush, opens a notification window for me, asking me what duration I want to do. There is an option of 3 minutes and 10 minutes. I will choose 3 minutes and click on 3 minutes. So letting me know that I need to load priming buffer into sample tube, which we have done. So good to click on OK. And then instrument flushing all the beads out of the cartilage and shouldn't take more than a minute. And then I'll show the rest of the steps. Okay, the 3 minutes flushing step has finished. And now we can load our sample to our sample tube. We don't have a real sample, but I'm just gonna click on OK and stop our demo at this stage. If you have, like, of course, cells, you can continue with analyzing your cells and sort and going through that. And but as I mentioned, I want to show you how to finish the work, do the cleanup, and how easy is that. Okay. At this stage, I would like to stop the instrument. So, because technically I'm pausing in the middle of the workflow, I'm going to close the window. When I want to close the window on the normal procedure, you have the stop button here that you can click on. But as I mentioned, I'm pausing the workflow. The instrument, the software, giving me a notification if I'm sure, because the data is not saved and gonna lost, which is fine for today. So I will click on yes. And then you hear that the instrument stopped and asking me to remove the cartilage and then close the door, which I can click on OK. And then it goes through a system change. What I'm going now is to remove my sheets from the sheet holder, remove my fax tube from sample holder, open the door, remove my priming tube, my waste holder. And by the way, there are some PBS in both of my priming and waste holder, which tells you that the priming goes through, I unclick the pumping and then undo all of this tubing. I'll do all of this tubing. And then when you want to remove the cartilage, you need to hold that lash up and then take your cartilage out. I'm going to put the cartilage back to its plastic container, and you can just dispose it according to your institute biohazard practice. There was nothing in there except PBS, so it's safe to put it on this surface now. And then it asked me to clean cartilage in the instrument manual if needed. And then I click OK. And then it asked me to turn the WOLF device off. I click OK and turn off the instrument, and the software also disconnected and closed, and pretty much you're good to go to your other experiment. And if the day is finished, you don't need to go through excessive washing steps that you're doing with your traditional sorter, and done. Well, I hope you enjoyed this demo. We can go back to Tom now, and he shows us some real results from samples he had. Thank you.
So from here, I can show some typical data, hopefully to add you to understand our platform. And as I briefly mentioned, we also have virtual demo data for cell line development. Actually, it's an involved process, but our scientists make it easy, and I can share that with you through Saba if you would like. So with that, I can share some data, and let's see. Let me share the screen. Okay, first I want to share two papers to give you systemic ideas about how we do things in terms of genomic sample preparation. This paper is published by Nicole, which is our scientist, and Nanocellect. There's a detailed introduction material and method, and even explain the mechanism of our microfluidic sorter, how it works, and how sample is being collected, and how PCT worked, and the WOLF or G2 play a role in the sample preparation for the chromium or the 10x, right? And until the SNP plot. And this is some of the typical data in terms of a median gain and median UMI, which I mentioned. And this is some of the graphics that the cluster plot. So those are the data that we covered today. And if you want, we have a detailed protocol, application note, and a publication on this topic for genomic sample preparation, particularly for 10x. CLD, we also have a publication from Donna. So this is high efficiency sorting and outgrowth for single cell cloning of mammalian cell lines. The cell lines she worked on is listed here. CHO cells, XP293F, hex cells, MCF, Jurkat cells, and A549. What difference of this paper from what I talked with you is that she listed the media that is being optimized for each of the cell lines. And so if you happen to doing those cells, you might reference to this this publication where you could use optimal media for your cell to grow. Because the difficulty thing for cell line development is that, okay, the cell normally living in a population, like in suspension or adherent, but now you are asking single cell to live in its own environment to grow into a colony. And that's a significant challenge for a lot of cells. To optimize media for that purpose to grow by itself is critical. So this paper describes some of the preliminary optimization for each of the cell lines. For example, the ES cells I mentioned in my previous presentation has a mixture of its original culture media, about 20%, with EX CHO cell media, plus some ACF, which is supplemental express the protein by genetic means. It's still complying with GMP or on the clonal modification side in terms of complying with the regulations. But this media is critical for the CHO cells to grow in a monoclonal way to 90% outgrowth ratio, just for your information. That's why we all compete with other competitors on top of the gentle sorting. And so these two papers are the backbone of the one that I talked about. Primarily, I want to mention for monoclonal demonstration, you need to follow the cells from the day of deposition to the clonal growth from a colony. And this paper also described that. And also, we're saying we're outbeat the limiting dilution by far with our machine's help. And this data show exactly how far we are improved based on the limiting dilution. For example, the K1 cells limiting dilution would be less than 20% outgrowth, where ours is reached 90% or something. And there are some other things you can look for. I just don't, but we have this data here. So with that, I will just cover that two application with a little bit scientific backing. Let me see, how can I close this one? And then I want to talk about the typical example of some of the actual data. Okay, one example I want to show you on the genomic sample preparation is the nuclei sort. I don't know any of you doing that, but this is a critical application for 10x, and it works beautifully on our platform. The reason is very simple, because the nuclei is very fragile, and in our system, if we keep the temperature right, like at 4 degrees all the time, and we can sort the nuclei intact and very efficient, because the nuclei can be stained very well by number of dyes, 7AAD, PI, or JQ5, JQ7. So it's a very optimal technical area where our machine really excel and doing very well. I just want to show some data here on this application. So this is one of the genomic application, which is could be used to 10x, of course. We use the 7AD to stain the nuclei, right? The nuclei is that would be no problem. We can stain the nuclei very well, and then we sorted it from there. Saba mentioned on the right hand corner there's a settings, right? And this settings typically we just leave it there for nuclei as cells. The setting we choose the cells, right? Not rainbow beads, not calibration cells. The shields we use PBS, but with BSA. BSA is to help the nuclei to keep intact, right? And the channel we use is FL3 because FL1 is typically for fluorescing, FL2 typically for PE, FL3 typically for red dyes like 7AAD, right? So we're gonna use the sorter channel to do sort and pick those nuclei. A typical gate on the scatter plot is look like that. Typically, the first gate you're gonna use is the forward scatter or back scatter. So this one would differentiate debris from the cells or the nuclei because debris are smaller, so they are in corner. So that's the typical gate, the first gate. The second typical gate, we said we can differentiate doublet, is use the forward scatter versus the forward scatter width. Because if it's a doublet, the width would be higher. It's something like here. We consider those as doublet. Of course, typically you need to go by your cells and how your cells behave. But generally, we use this as a second way to screen out the junks. So first, to screen out the debris by selecting the forward and back scatter. And secondly, we pick the singlet from the doublet by applying the nuclei gate to this one. And this gate, which is singlet, is a subpopulation of the whole population, will be applied toward the third scatter plot, which is the FL3, which is 7AAD versus the backside scatter. So this, the third one is, okay, this singlet has these three populations. We're gonna pick up the middle one, the one stained with 7AAD. We're not gonna pick a subpopulation with too bright. This is the typical one that stained with 7AAD, that is the nuclei we want to isolate. And then we just sort from there. Okay, and then whatever, how many cells we sort, that's how we typically do. We gave them out and we pick the one that we want. So that's the typical data. Let's see whether we run these samples. This is just more data to show the post-analysis reach the 84.5% from 64%. Of course, the people sometimes say, oh, post-sort is 90%, but it depends on sample preparation and how things were done. But in this case, that's what we got. Concentration-wise, we sorted as, for bulk sorting, we normally recommend 1 million cell per mil. So in this case is 900 or 0.9 million cells per mil. That's a pretty optimal concentration. In 19 minutes, we sorted 57,000 cells. So that's more than enough for a typical 10x Chromium sample input. Okay. There are some other things that we report to the customer in a typical setting, but that's typically the data we use for 10x. The other example I want to point out is for cell line development. Let's see. Give me a second. To sort certain cells into 96 wells. So that's typically for cell line development because we need individual cells to be able to grow into colony, which is pure in terms of cell lineage. And that's what we do. The experiment one is this situation. Typically, like any other flow, let me see if I can. is you first gated the forward and side scatter for the cell population. And from there, we typically gate a singlet population. In this case, we pick the live cells stained by 7-AD, right? Not the dead cells, like the nuclei. Well, the nuclei is not dead, it's just stained like that. So the live cells, and then we deposit that into 96 WOL. And this is what typically show as the data. Let's see. There's the second experiment is iPSC, isolate the GFP labeled iPSC or GFP labeled cells. There's another typical scatter plot for first cells. This is to get rid of the debris, and then singlet, and then the GFP. Here we which is the higher expression of the GFP, right? Only the top 23% of the expression. The high pressure is on on this X-row. The more toward the right, the higher that it is. Okay, this is only backscatter to point out to the cells. So in the box sorting, we ask it to sort how many, and the instrument would be able to do that for us. This is a deposition into 96 WOL. So I just think those ought to be the typical data that you would experience during your typical sorting process. I don't, right? I mean, yeah, that's all I want to share with you. Sorry, my background is I'm sitting in a hotel, so. If you have any questions, you may ask. And also, I want to give me a second. I want to point out one. I don't know how it's gonna play here. There's too many windows open. Let's share screen. Okay. With Saba's demonstration, I'm not sure it's necessary, but we do have a single cell virtual demo system for you to see. Give me a second. Sorry, I may not be able to play this, but sorry, I'm not gonna share this at this point because I don't know whether you need to learn that or not, but we do have this video available if you like. For at your spare time, you want to look how the cell line development being worked out, or how her publication, Donna's publication being worked out for this purpose. I'll be more than happy to share it through Saba if you like. But anyway, if you have any questions, let me know. But
thank you so much, Tom. I know that was not in the plan, and
right.
But that's all good. If anyone is interested to watch the video, let me know. I'm happy to share the link with you. Thank Tom for your time today. Really grateful for having you today.
It's a great honor to see you guys interested in this. Very happy to help out, but it's a great opportunity for me to be here. So
on that note, this is technically the end of our seminar and demo. If you have any other question or you would like additional information or any application notes, please let me know. Feel free to reach out to me. I'm more than happy to answer further question if required.
Yeah, I'd just like to say thank you to Saba for running this demo today. It's always challenging when we do things virtually, but given the case numbers, we felt this was the best way to do it. Hopefully the manual movement of the camera wasn't too nauseating for people, but just a really big thanks to Saba. I think it went very well, and I hope everyone got something from the demo, and thank you also to Tom for your very knowledgeable input and the really great case studies. So thanks very much.
Thank you, everyone. Have a lovely rest of the day, and see you soon.
Thank you, Saba. Thank you, everybody. Bye-bye.
Bye.